So what's the deal with type 2 diabetes these days? It seems like you hear about it all the time, almost as if it's a pandemic. And you've probably heard this idea of too much sugar and then you have to take insulin. What's going on there? Is there something wrong with our cells? Is there something wrong with what we're eating? What's the root of it? Well, in this video, we're going to talk about how cells go through signal transduction, and we might get to the root of the problem. What causes diabetes? Let's get started. So in the last video, we looked at how cells communicate with one another. And I mentioned that cells receive signals. So in this video, we're going to go through the entire process of how cells receive signals and respond. So the first step in how cells communicate is they receive a signal. So signaling begins with the recognition of a chemical messenger. Now this word is pronounced, I've heard several different pronunciations, ligand, ligand. I'm going to go with ligand. You can, I guess, say it how you want it. So a ligand basically is just a chemical messenger that's going to be received by a receptor protein. All right, so here we've got a cell, right? You see the membrane. Here is a receptor protein, right? So there's some sort of receptor protein. Now, right now it's inactivated. But when a ligand is received, right, a chemical messenger is received, that activates a process called transduction. That activated enzyme or receptor then is going to start a series of events, and ultimately that will lead us to some sort of response in the cell. Now, that response can be many different things. It often includes something like transcription, but we'll go into those ideas in a moment. So, stage one of this whole process of cellular communication is all about reception, receiving a signal. Now, when that happens, when the cell receives a signal on its receptor, there is a conformational change, right? It conforms, it changes its shape in some way to start the transduction. And there are several examples of receptors that I'll go through here, but basically what you need to understand is this. Originally, before the receptor is activated, it's in one conformation. Once the signaling molecule or the ligand joins, it will change its conformation. In this case, notice, it sort of opened up this channel to let some ions through. So the binding of the molecule creates a conformational change. It changes the conformation or the shape so that something happens and transduction will occur next. Now, I'm given you right here several examples of types of receptors, right? So these receptors here, G protein linked receptor, ligand-gated ion channel, which you kind of see here, and receptor tyrosine kinase. So the next three slides I'm going to go through will go through these three receptors. Okay? So the first receptor, one that gets a lot of press, it's involved in a lot of things, is what we call the G protein linked receptor. Okay? So we're just talking about the receptors here in the membrane that are going to receive the ligand. And so what you see here is a G protein receptor, GPCR. And what that does is when the ligand binds, right, this G protein, which is here attached, will move and activate this particular subunit to come over here and activate this enzyme. This happens to be adenylyl cyclase, you know it's an enzyme, ASE, which then starts another cascade, and we're going to eventually get some cellular response, okay? So the G protein linked is called that because it's got this little G protein right there that's linked to the receptor that will then move over and activate an enzyme so we can get some more process to occur, okay? So the first receptor is the G protein linked receptor. We'll go through some more of this later. This is the second and third parts of cell communication. But the second receptor I wanted to mention is ligand-gated ion channels. And so how this starts is you'll see that, all right, we got a cell membrane and we've got a closed channel, right? No ions can move through. When the signaling molecules bind, that creates, again, a conformational change so that these ions can move through, okay? So the signal was, let the ions in. So we call this a ligand or ligand-gated ion channel because it's a channel that ions are going to go through, but it's gated, it's controlled by the ligand that binds. That's the second type of receptor. The third type of receptor is called a receptor tyrosine kinase. And you may remember kinase means a phosphorylating enzyme. So we're going to see some phosphorylation occur. What you see here is a receptor tyrosine kinase that's in its inactive form. Okay? Well, then we're going to have a signal molecule, a ligand. It will come, and when this molecule binds, it signals for these two pieces to sort of come together, right? Notice how it went from two separate units to one unit here. So then, once these come together, it's going to start this kinase or this phosphorylation activity. It's going to stimulate that. So now what you'll see is the phosphorylation has occurred. Phosphate groups. The tyrosine down here has become phosphorylated and it's activated, right? So this is self-phosphorylation. It's an autophosphorylation process. And then, of course, that signal, once this happens, will activate some signal proteins to carry the messenger into the cell to do what we need, okay? So this is the third type of receptor that I'm going to talk about. Receptor tyrosine kinase. It works by taking these two singular units, bringing them together when the ligand binds, which then starts this phosphorylation, which sends a signal. Okay, so those were basically, that was all stage one, the three types of receptors. We now want to move into stage two. Once the receptor has bound, right, or it came down, the ligand has binded or bound to the receptor, what we're going to see here is that transduction needs to occur. So what transduction does is it converts the signal, right, we receive that ligand, into something the cell can understand and get a cellular response. And this normally works as you see here, right? So the ligand binds to the receptor, which activates this protein, which activates that protein, which activates that protein, which maybe comes in and leads to, I don't know, transcription or something in the cell, right? Where we take a piece of the DNA, make a copy of it into RNA. Other times, right? This is another example, right? I'm showing you several different examples here. This activates this, which activates, phosphorylates this, which together with another thing that's already in the cell joins and then comes in and serves as transcription factor or something. And then another thing that might happen is a ligand binds. It starts what we call a phosphorylation cascade, right? Where one thing phosphorylates another, which phosphorylates another, which phosphorylates another, and it starts this cascade. So many of these signal transduction pathways will include this phosphorylation. You see all these phosphate groups. Because those are basically a series of protein kinases, phosphorylating enzymes, that phosphate to the next one, so you get a cascade. So this activates that, which activates that, which activates that, right? And the nice thing is, once this is activated, it can activate more. And then these can activate more. So basically this results in what we call some amplification. So the transduction of the signal is stage two. After the ligand is bound, this is where we're going to get some different processes happening in the cell so that eventually we can get the cell to do what it wants, right? It would just be easy if the cell understood English, right? And we could say, okay, what we need you to do is transcribe this gene. But that's not how cells work, right? They don't speak English. What they do is they speak chemical, I guess you could say. They need different chemicals to be activated to tell them what to do next. So that's stage two. So as you probably noticed, once the ligand bound, there were lots of things that happened inside the cell, sort of like secondary messengers. Secondary messengers, as you can see here, right, we got a lot of messengers. These are essential to the function of the cascade inside the cell. So if you think about the ligand that bound, that was what we might consider the primary messenger, or the first messenger. These that are inside the cell acting are still sending messages, but they're kind of like secondary messengers. They weren't the first, but they're kind of secondary. So there are several examples of secondary messengers that you're going to see. Cyclic GMP, the one you see here is cyclic AMP. You also see some calcium ions, right? Those are the ones I've shown you in this example. But inositol triphosphate is also a secondary messenger. The whole point of secondary messengers is really just to continue the functioning of the cascade. So the whole thing started when the ligand bound to the receptor, but we need the stuff inside the cell now to continue as well. What you're seeing here happens to be CREB cascade, and this is just a transcription factor. So this is the specific series or pathway to activate CREB, which is a transcription factor. And these secondary messengers, like you see here, were essential in that process. Then finally, stage 3, response. So once the signal passed through the pathway, we're going to need to get some response. That's the whole point of this. The cells are communicating. It sent a signal. Now we need a response. And they can be in many different ways. I've showed you this diagram before, but in this diagram, what you can see, the ultimate response was transcription from the DNA into some messenger RNA. Okay, that might be a response that we wanted. Another is that, we need some enzyme in the cell to start working. Or, start rearranging the cytoskeleton because we're going to divide the cell, mitosis. Maybe we need this gene activated or turned on. Or maybe we need the cell to undergo cellular suicide or apoptosis. So cells need to be able to do lots of things, and they need to know when to do those things. So the cellular communication is important because we can get all of these different responses depending on the ligand that binds to the receptor. Now, as is common in biology, things can go wrong. So what happens when this whole signal transduction pathway that we've been talking about is blocked or is defective? Well, the conditions can be pretty bad, quite deleterious, or actually it can be preventative to stop something from happening. So there can be lots of times that these pathways don't work. Sometimes it causes disease. Sometimes we use drugs to actually stop some of these pathways. So some examples are obviously cancer, diabetes, which I led the whole discussion with today. Neurotoxins work, antihistamines block this pathway as well. I'm going to give you the example to sort of wrap up how we started the whole thing with diabetes. So diabetes is all about, right, too much sugar, too much glucose in the blood. Well, how glucose normally works is that you've got a hormone, specific protein called insulin, that binds to a receptor on your cells, on a few of your particular cells, which starts a signal cascade we've talked about, activates these GLUT4 proteins to take glucose and bring them into the cell, right? So if you've got too much glucose in your blood, you need to bring it out of the blood, store it in the cell. Insulin tells your body to do that, or your cells to do that. Bring in the glucose, store it as glycogen in the liver or something like that. Now, if this pathway goes bad, either the receptors become resistant to insulin, right? You might have heard of insulin resistance, type 2 diabetes. So if the receptors become resistant to the insulin, or for some reason the particular cells in your pancreas stop making enough insulin, then this whole process can't work. You can't take glucose out out of the blood, put it in the cells, and store it. So when these pathways have changes, they can be quite harmful. So cells communicate. That is the point of this whole video. And we talked about, just as a reminder, the three things that happen. The first, we have a receptor that is activated by a ligand. Second, the signal is transduced or changed into something the cell can understand. And then finally, the third thing that happens is that we get some response, often something like transcription, but it could be many things that happen in the cells.